Hello everyone! Did you ever think it was possible to fool your own brain with a Trojan horse? Well, it turns out you can. I am Lieve, a researcher in the field of biochemistry and drug development. At Biochemistry Explained, we make scientific literature accessible. The topic we are going to discuss today is sneaking therapies into the brain. As you may know, the brain is surrounded and thus protected by the blood-brain barrier, also known as the BBB. A recent paper has discussed a method where they used an IgG antibody as a carrier for both an BBB receptor binder and a therapeutic cargo molecule. The BBB presents itself as an immense roadblock to many drugs, especially larger ones trying to get into the brain. To overcome this, researchers have tried to use the endogenous receptors present on the barrier, of which the transferrin receptor is the most studied. This receptor is normally used to transport iron across the BBB. The strategy in which you use an endogenous receptor to get into the brain is called receptor-mediated transcytosis. Previous attempts of a brain shuttle involving an antibody have been made. However, what is unique about the approach used in this paper is that the transferrin binding domain was incorporated into the antibody structure instead of being attached to it. This provides the unique ability to create a bispecific vehicle since two antibody arms are free. The researchers attach an antibody to the vehicle that selectively targets both BACE1 and tau, two sources of pathology in Alzheimer's disease. BACE1 is an enzyme crucial in the formation of beta amyloid, the main component of amyloid plaques seen in the brains of Alzheimer patients. Tau is another protein found in Alzheimer, which aggregates and damages the neuronal network in the brain. Kariolis and colleagues showed that by administering their bispecific antibody targeting both BACE1 and tau, all targets were bound and also both BACE1 and tau were inhibited in cell culture. After successful testing in cell culture, they tested their transport vehicle in mice. Mice treated with the antibody connected to their transporter vehicle showed 40 times more anti-BACE1 antibodies in their brain compared to the mice that simply received the antibody. A 30-fold increase was observed in monkeys, clearly showing that the vehicle molecule improved BBB crossing. Amyloid beta showed a 2-fold drop when using the transport vehicle, while standard anti-BACE1 showed no explicit pharmacodynamic effect. This illustrates that anti-BACE1 is still able to exert its function, even when it's administered connected to a transport vehicle. By the way, if you don't know what pharmacodynamics is, or if you don't know any of the other terms I mentioned in this video, let me know in the comments below and I will also post some links with some extra explanations. This study shows that it is possible to use a transport vehicle, in this case an antibody with a BBB binding domain, as a Trojan horse to sneak therapies into the brain. As always, future research is required, especially since this was a study done on mice and non-human primates. This requires more translational approaches in future studies. This does not take away that this is a really cool study to try and get therapies into the brain and reach their target. The importance of this is understated, since many neurological disorders, such as Alzheimer's disease, remain largely untreated at the moment, which is a huge burden on society and requires us to find better and more efficient drugs in the future. During my time as a student and researcher, I have come to the conclusion that scientific literature often does not reach a broad audience. However, we want to change this by making short and informative videos about novel findings in the field of biochemistry. I hope you enjoyed this video. Please hit the like button and subscribe. If you have any suggestions for future videos, please let me know below in the comments. See you next time!